Hi, my name is Lauren Albrecht, and I am a postdoctoral researcher at the University of California, Los Angeles. And today I'm going to tell you a little bit about my work. And I'd like to start with one word, methylation. Now, I first learned about methylation as I was studying sudden death in young adults in a disease called arrhythmogenic right ventricular cardiomyopathy, or ARVC. Now, you may have heard of this disease as it is frequently in the news, where young, healthy athletes die suddenly during practice as a result of cardiac arrest. Now, these patients have no prior health conditions, but they present in the hospital with enlarged hearts that have transformed into a fibrous fatty tissue. And now this disease is very mysterious, and there are currently no treatment strategy options. So in my work, we wanted to look more at this at a molecular level. And to do this, I designed a new genetic sequencing technology that allows us to visualize our genetic code, our DNA. And what I found was very interesting. A single mutation occurred in patients with ARVC at a residue that is very important as it was modified by methylation. And at this time, there was actually no previous evidence linking methylation to ARVC, but I had a gut feeling that there was something potentially important here. So to give you a little background, methylation is a chemical modification that occurs in every cell of the human body. And it acts kind of like a light switch, an on-off. And it occurs on DNA and on protein that DNA is encoding. And upon scouring the literature, I quickly discovered that one of the reasons why methylation has been overlooked is it because of the lack of sensitivity in detection systems that are currently available. So I applied a new technique called electron transfer dissociation mass spectrometry and found that in fact, the loss of methylation occurred in ARVC with this mutation. And this recapitulated what is seen in human disease in using our animal models, and together revealed a new important role for methylation in cardiac dysfunction and in normal cardiac health. And so methylation is not only misregulated in the context of this rare genetic heart disease, but is actually implicated in diseases such as cancer and diabetes. And my current research is looking at cancers where methylation plays a central role in two of the most lethal types, which are colon and liver. And in these cancers, methylation is out of balance, where it's typically upregulated or increased. And in fact, some of these cancer cells, they may actually require methylation in order to proliferate. And thus, this provides a very interesting idea that perhaps blocking methylation could be a novel way to interfere and to disrupt tumor growth. However, current limitations exist for targeting methylation as it's so important for the human body. So we need to understand how methylation can be specifically interfered with in these contexts. And as I was conducting these studies, a series of very compelling reports came out from other labs that showed that methylation inside of our bodies can actually be changed by our diet. And this is because methylation is acquired from a metabolite that we consume, an essential amino acid. It's called methionine. And methionine is in a bunch of different foods that we eat, where Brazil nuts, for example, have some of the highest levels. And I thought this was very interesting, as one of the very unique qualities of cancer cells is their ability to sense their metabolic environments and adjust to the available nutrients as they grow and expand exponentially. And the concept that diet can be used as a treatment to decrease cancer has been very controversial over the past few decades, but there is currently a revolution occurring in our understanding of metabolism inside of the human body, as there has been increased sensitivity of instrumentation and new live cell imaging approaches. So I wanted to utilize this new base of knowledge to ask a very simple question. If we block methylation through diet, through decreasing methionine, can we block tumor growth? And so I used classic cancer survival assays and found that in these cultured liver and colon cells, if we remove one single nutrient, if we remove methionine, this was sufficient to decrease cell growth and tumor growth in animal models. It's altogether showing that if we can reduce methylation through diet, through methionine, that this could be a new additional approach to complement current chemotherapeutic strategies for these patients. Altogether, my long-term vision is to design and to implement new tools to modulate methylation in different disease models and in clinical settings. And as methylation is a fundamental aspect of human biology, one of the greatest challenges in successfully targeting methylation will be to understand how we can achieve tissue specificity for precision medicine. But by designing new tools to interfere with the molecular basis of methylation, this line of research has the potential to impact the diseases that plague our society most.